to the organizers for this invitation to speak with you this morning. So I had a short list of goals here. We only have 10 minutes, and I would love to, we can take 10 hours to talk about triple-negative disease and hopefully come up with some new options. But I wanted to go over just a bit about some of the basics of triple-negative breast cancer to give us a foundation for what we know and a lot of what we don't know, and talk a little bit more about how we're learning more about different subtypes of triple-negative cancer that are really helping us focus our treatments and our options for treatment based on trying to be specific and smarter about the tumor that we're dealing with. And then lastly, give just a handful of examples of some of these important pathways in triple-negative breast cancer that we're being able to effectively work on and target. And also use this as an opportunity to share a little bit of our work and work around the country on triple-negative disease that we're very excited about. So, just to start here, as those of you who may live with this disease know, triple-negative breast cancer is that cancer which lacks all of our targets. And someone very wise once told me, if you're calling it by what it's not, i.e. triple negative, it means you must know nothing about what's going on. And I have to say, unfortunately, there's a very strong component of truth to that. However, I can say that also that within the last 10 years that I've had the privilege of working with patients, I think that we're making such huge strides and we're coming a very long way. And so even though we still give it this baggage term of triple-negative disease, I'll show you that we're really teasing it apart and coming up with very rational strategies to target this cancer. By definition, triple-negative breast cancer is breast cancers that lack the estrogen, progesterone, and HER2 proteins or overexpression. There's terms that get thrown around a lot. A lot of triple-negative breast cancers are called basal. That's one of the main types of triple-negative. But the two terms are not actually synonymous. There are some triple negatives that aren't this basal-like phenotype. There are other features of the tumor that the pathologists help us look at. Triple-negative breast cancers tend to grow very rapidly, so if on your pathology report, they'll talk about the grade of the tumor, which often is a high grade. They'll talk about the Q67, or the rate of proliferation of the tumor. And triple-negative tumors in general are known to be the bad actors that are growing quickly and of a high grade. We know that in individuals who harbor germline mutations of the gene BRCA1, these are patients who are more likely, we tend to see more often, they tend to develop this triple-negative type of breast cancer or the basaloid subtype. In terms of how these tumors react, how they act and behave, they behave badly, pretty uniformly badly. We know from many reports that individuals, patients who are diagnosed with triple-negative disease tend to have a higher risk of the breast cancer coming back after successful surgery, and that outcomes, when oncologists talk about outcomes like disease-free survival and overall survival, we know that we have a lot of work to do for women with this type of breast cancer because their outcomes tend to be poorer than what we see for women who have the HER2 positive type and for hormone receptor positive breast cancers. There are certain features of triple-negative disease. We tend to see more spread to vital organs like the lungs, the liver, and the brain. And sometimes we also see that these tumors tend to occur more commonly in younger women, sometimes in African American and other ethnic populations as well. The funny part of triple-negative, for all of these very sad things and difficult things about the disease, when we talk about our chemotherapies as our poisons, the triple-negative breast cancers, those are the cells that actually tend to respond best to our cytotoxic chemotherapies. Because they're growing fast, they really are the cells that are growing fast, so that when we do give you the poisons, we can kill those cells pretty effectively. The hard part is that they're awfully damn smart tumors as well. And so we treat and we can kill 99% of them, but there's a few cells that are always smart enough to be resistant. And then those are the cells that come back and we repeat the process over and over. So we have a lot of work to do, and there's been a lot of really phenomenal scientists who are helping us understand more. This is something that will look very busy. This is called a genomic heat map, and this is some really seminal work that was done by the group at Vanderbilt. And when we talk about triple-negative, like I said, what is that group? What does that mean? If we start to dissect and look more closely at the tumors, we can look at the gene profiles. So we basically sequence all the genes in the tumors and try to say what genes are up, what genes are overexpressed, what genes aren't functioning at the same level. And what we've been able to do is really take the triple-negative, which is already a small portion of breast cancers, roughly 15 to 17%, and we've been able to break them down now into probably 6 main groups. So this is good because we want to know more about the tumor and focus our efforts, but it also makes some things more difficult because now when you talk about triple negative, we really want to know maybe what type it is, and that also makes it a little more challenging when we think about doing clinical trials and things because we're taking smaller and smaller groups of women who we'd like to be able to design specific trials for. So just to kind of summarize, this is a map looking at the way different genes are expressed in the tumors, but we can identify now that most triple negatives, the biggest group are the basal type, and these again are the types of breast cancers that develop in women who harbor BRCA1 or 2 gene mutations. There's a type of breast cancer that Dr. Desis referred to, the immunomodulatory. These are really exciting because these tend to be the triple negative breast cancers that have the highest influx or the highest percentage of these T cells and immune cells that can help us generate immune responses. And then there are a couple more applications, mesenchymal-like, and these I think are still the ones that we absolutely need the most work on. We know that there are certain gene pathways that are upregulated in these, and there's a lot more work to be done. These again are very rare, but very difficult. And then the last group that I'll spend just a little bit of time talking about is that among those tumors that we say don't care about hormones, there's a type of triple negative called the luminal type that actually appears to be somewhat dependent on androgen. And androgen is a male sex hormone, and so there's a lot of excitement about trying to think about targeting these, and these kind of behave more like estrogen receptor positive tumors. So I hope you can understand, I think we're going to do better as we learn more, but it just also makes things a little bit more complicated. I should say that this type of breaking the tumors, the triple negatives apart, is not something that we routinely do yet in clinical practice. We can make guesses about it based on how the tumor behaves and about certain features, but this type of genomic testing is not quite to prime time yet. So please, we think about it, but we don't necessarily test for it in a way that we can always tell you exactly which type you might have, but we're working there. So I'd like to give some examples of some successes in some areas that we're really excited about. So for these basal-like triple negative breast cancers, which are a big component, we know that they have some inherent difficulties with repair and damage to DNA. And there's a whole class of drugs that have been developed for ovarian and now are being studied in breast cancer that help leverage some already present defects in these triple negative breast cancers. So they're a class of drug called Parp Inhibitors, and Parp is a pathway that helps repair damage that happens to DNA. And if you get too much damage, then that causes the cancer cell to die. But if you just keep accumulating mutations, the cell will try to keep fixing them, and sometimes that just begets more mutations and begets more aggressive cancers. But Parp inhibitors have shown disease activity by making the cell get so damaged that it dies, in an easy way to say. We've been really fortunate, and one of my colleagues, Eve Rodler, who is here and is now in San Diego, we got to work together on one of the first trials with a Parp inhibitor for patients with triple negative breast cancer. And this is just what's called a waterfall plot to show you some of the responses. This was a combination of an oral Parp inhibitor with 2 types of chemotherapy, one of which is cisplatin, which is another chemo that's really good at damaging DNA. The idea is to put these drugs together so that you get more DNA, fatal DNA damage. And these results were in a phase 1 study, again, of patients who were heavily pretreated. And what we saw is that a good number of these patients had very good and some nice longer-term responses. And the patients who seemed to have the best benefit with respect to having their disease go away to the greatest extent were those patients who harbor these BRCA1 gene mutations, which we think puts them in a better stead for the use of Parp inhibitors. So this type of DNA damage and using Parp inhibitors is something that's being studied in a number of different settings. And some of these trials are for patients with early stage disease, but this is also efforts really being leveraged across different stages for women with triple negative breast cancer. Eve's study, based on the work that she did here, is now happily a national trial that is open. That's a phase 2 study where we've taken the results that we generated here and are now proposing those forward for a very large trial. And this will be for patients with advanced triple negative breast cancer who are going to get a combination of the chemotherapy that damages the DNA with or without the oral Parp inhibitor. And we're very excited about this trial as well. Parp inhibitors are also being studied because of their differential or hopefully better effect for women with germline mutations. The Olympia Trial is a large cooperative group study that's looking at giving these drugs after a woman completes her early phase breast cancer treatment to see if we can help these women do better. And so this is an area that we're very excited about in terms of DNA damage and Parp inhibitors. These are not drugs that are yet approved for breast cancer, but are coming along, and we're very hopeful about. Okay. I am over time, so I'm going to talk really quickly about androgen receptor. These are the type of breast cancers that have the androgen receptor, which makes them potentially function a little bit more like a typical hormone receptor positive breast cancer. This is another small percentage of triple negatives, but these cancers tend to grow a little bit more slowly and are potentially targetable. And Dr. Linden, my colleague, has a great trial open with drugs that are targeting the androgen receptor that are oral medications. And we also are working with TBCRC, one of our breast cancer research symposiums, to design clinical trials that are targeting this particular population of disease. I'm going to skip through this because Dr. Desis did a wonderful job, but one of the most exciting things, again, is the use of these checkpoint inhibitors, which essentially are these drugs that unleash the immune system and release the brakes. She talked with you about pembrolizumab, so I'm going to skip through this. This is one of the quintessential drugs. And to build on some of the work that she showed with the initial phase 1 study, we're really thrilled to be opening a trial where we're combining the chemotherapy with the immune checkpoint inhibitor for patients with triple negative disease. And so we're trying to build on that initial study that she showed you with pembrolizumab alone to see if we can get better and longer duration responses for women with triple negative disease. At the bottom of the slide, I'll show you very quickly, through our phase 1 group, we have the privilege of working with a lot of different opportunities for phase 1 combinations of these immune therapies. Again, the idea is to try to build on using the checkpoint inhibitor blockade and add other drugs that help stimulate our body's immune response to fight the cancer. And this is just a truly fundamentally different way of killing cancer. We're not actually doing anything to the cancer cells, but we're helping our body's immune system to identify and attack and kill the cancer. And this is something that I can't tell you, for those of us who treat breast cancer, this is like a paradigm shift that's nothing short of truly amazing. Okay, the last thing, and I promise I'm sorry for going a little over. So if we continue on this scheme of immune therapy, the drugs that I showed you are drugs to try to help our body's own immune system fight cancer. Well, what if we do it in a slightly more aggressive way? And one of these ways could be taking T cells from our patient's blood, genetically modifying them so that they specifically attack the cancer. So essentially you pull out T cells from the body, you change them in the lab, and you make them into warrior T cells that go exactly where you want and kill the cancer. And this is an approach called adoptive T cell therapy that's been used with quite good success for individuals with liquid types of tumors, leukemias and lymphomas. And in collaboration with investigators here at the Fred Hutchinson, this is work by Stan Riddell and his colleagues, and work that I'm privileged to work on with David Maloney and their team, they've identified a protein that is present in some triple-negative breast cancers called ROR1. And it's a protein that seems to be primarily expressed only on the tumors and not so much on normal cells. And so what the idea is here is that Dr. Riddell's lab has been able to generate a way to modify a particular woman's T cells to attack that ROR1 protein. And by giving those T cells back to the patient after you change them, infusing them like a transfusion, the idea is that these warrior T cells are going to go directly to the tumor and kill it. Now this is a very, very new and actually truly exciting, but very new and very risky new way to treat breast cancer. And we are very pleased to have this trial open now for patients with advanced triple-negative disease who may be candidates to do something that is very much completely different. We hope will be very successful, but also comes with a fair amount of risk because, again, this is very brand new. So it's exciting to be here on the cutting edge of some of these therapies. And again, I'll stop here. Sorry for going over. Thanks to our amazing scientists here